This lecture is on alveolar ducts and alveoli. So, the respiratory bronchioles, I hope you remember them. Those are the finest branches on the bronchial tree. The respiratory bronchioles then open into expansive chambers called alveolar ducts. And the blind pockets that open into these chambers are called alveoli, and these, the alveoli then, are where gas exchange finally takes place. So we have these chambers then, and then off of them we have all of these little bubbles, like so. So each one of these little pockets is made out of soft tissue. They expand. When air enters them, they contract and push the air back out again. And the walls of the alveoli then, those are where gas exchange actually takes place. So here then we have the alveoli. So each lung contains approximately 150 million alveoli, sometimes known as alveolar sacs. And this is why the lung has a spongy appearance. As I've said before, the point is that your lungs are not balloons on the inside. They are not simple balloons on the inside. They are masses of tiny, tiny balloons, all of which have their own separate air ducts getting the air to them. And so there's a lot of plumbing, a lot of air passages involved, finally getting, as I said, down to there. So it means that your lungs are actually filled with these tiny alveoli, so that if you look at a sample of lung tissue, it looks kind of spongy. So alveoli then have no layers of of muscle or cartilage. And they are very delicate. So this is why you have all of those mechanisms to moisten the air, to temperature adjust the air, and so on, is so that by the time the air gets down to here, you're not going to freeze these little things. You are not going to bake them. You are not going to dry them out, and so on. So, as I said, these are very delicate tissues at this point. Now, additionally, there are septal or surfactant cells that are scattered among the linings of the alveoli. And surfactants, I hope you remember them, aid with decreasing and equalizing surface tension in alveoli. So, as a result, the alveoli don't collapse like burst bubbles, but they can be inflated. Just not too much. And then roaming, this is different now. We're now off of surfactants here. We also have roaming alveolar macrophages. You remember phagocytosis? Basically, where one cell eats another cell. So we have roaming alveolar macrophages, known as dust cells. And these roam the epithelium. So keep in mind, down at the level of your alveoli, the linings there of the alveoli are still going to be epithelial tissue. And so there, in that epithelium, we have these alveolar macrophages. So they roam the epithelium and they phago, that's a G, they phagocytize then. Any particles that are fine enough to reach the alveoli. So what did I just say there? Again, remember phagocytosis. This is where you have one cell that basically devours another cell and takes it in and digests it. So as I said, we have these alveolar macrophages that will go around and try and find any particles, like bacteria, for instance, that might have gotten that far. And crucially, do not forget this bit. We're now done with alveolar macrophages, but each alveolus is surrounded by capillaries. This is crucial because in order for gas exchange to actually be of any use to us, we have to be able to get the blood from our lungs past the respiratory surfaces and into our bloodstreams. So, as a result, each single tiny alveolus then has capillaries, tiny, tiny, tiny blood vessels all around it. Capillaries are your smallest blood vessels. We'll talk about them when we talk about the circulatory system. But they are also the level at which gas exchange takes place. So we have these capillaries surrounding the alveoli, and that allows the oxygen to go from the air in the alveolus across the alveolar wall into the capillary, so that now your blood gets new oxygen. And meanwhile, the blood in the capillaries is giving off the CO2 through the capillary wall, through the alveolar wall, and then into the air in your lungs so that you exhale the CO2. And that is that for this lecture.